Hello, everyone, and thank you for joining us today. My name is Dr. Susan Manganaro, and I'm a Senior Medical Director in Global Medical Affairs here at Rocket Pharmaceuticals. It's truly a privilege to welcome you to our session, The Critical Role of Genetic Counselors in Gene Therapy for Inherited Cardiomyopathies. Today's discussion will explore the evolving interface between genetics, gene therapy, and the essential work you do as genetic counselors in supporting families facing rare disease diagnoses. We have a robust set of speakers for today's webinar. I'll be opening the session by framing the current landscape in inherited cardiomyopathies and gene therapy. Dr. Jonathan Schwartz joins us. He's the Chief Medical and Gene Therapy Officer at Rocket Pharmaceuticals, where he leads clinical and translational efforts across the gene therapy pipeline. Since joining as founding CMO in 2016, he's played a key role in advancing Rocket's clinical programs and will be speaking today about gene therapy and the proof of concept in inherited cardiomyopathy. We're also pleased to have Cara Barnett. She's a licensed genetic counselor specializing in cardiovascular genetics, hearing loss, and implementation of genetics service delivery models. She's worked in both pediatric and adult academic medical centers, with her research focused on further defining cardiovascular genotype-phenotype correlations and access to genetic services. She's passionate about expanding access to genetic counselors through collaborative telehealth partnerships and is a current member of the NSGC Cardiovascular SIG, as well as the NSGC Expert Media Panel. She serves as the biocurator with the NIH Clinical Genome Resource. Cara will offer practical insight on how you, as genetic counselors, are uniquely positioned to guide patients through this complex and evolving space. To ground our discussion, we've set two categories of learning objectives. From a knowledge standpoint, we'll describe the genotype-phenotype relationships in inherited cardiomyopathies, outline current and emerging gene therapy strategies, and reflect on how these developments are reshaping the genetic counseling role. From a practice standpoint, we'll look at real-world considerations, how to recognize patients who may be gene therapy candidates, support their decision-making, and navigate clinical trial eligibility. So you might be asking, why now? Why are we holding this session? A recent study found that nearly 60% of genetic counselors say gene therapy is already impacting their daily work, and over 90% believe genetic counselors should feel confident discussing these treatments with patients. Yet 83% are asking for more training, so today's webinar is designed with that exact need in mind, to bring the science and practical aspects together. When we think about cardiomyopathies, it's important to recognize we're not just dealing with one disease, but rather a family of conditions, often with overlapping features and increasingly genetic underpinnings. In fact, up to half of the cardiomyopathies, including dilated, hypertrophic, and arrhythmogenic types, have a heritable basis. These are often driven by single gene mutations, as you can see here, and with overlapping clinical phenotypes. So it makes it even harder to rely on just imaging or symptoms alone for diagnosis. Each of these has their own completely different implications for prognosis and treatment. So that's why it's really important to have genetic counselors to help translate the phenotype into that molecular diagnosis and then enable patients to access not only tailored clinical management, but emerging gene-based therapies. As we continue to talk about the future of gene therapy, it's helpful to recognize where we are today. To date, the FDA has approved over a dozen gene therapies, many of which use AAV vectors, the same platform being explored for inherited cardiomyopathy gene therapy. You can see in the table here, these approved therapies target a variety of conditions from inherited retinal disorders to neuromuscular to hematologic conditions. But what's notable is that each and every one of these approvals has been for rare monogenic diseases, where the genetic cause is well characterized and exactly the landscape that we're going to be discussing today with genetic cardiomyopathies. Interestingly, there's no gene therapy that's been approved yet for cardiovascular indication, but the scientific principles and regulatory pathways are already in place meaning that gene therapy for these conditions is no longer theoretical. It's here and rapidly evolving. So for genetic counselors, this shift is significant, meaning that the conversations you're having with families right now are expanding from diagnosis and surveillance to include potential targeted treatments. Understanding which therapies are approved, how they work, and where cardiomyopathy fits in is part of staying ahead of this curve. As more gene therapies enter clinical trials, targeting well-defined monogenic causes, your ability to identify the right patients at the right time, help with result interpretation, and guide decision-making becomes even more critical. As investigational programs continue to move forward, targeting genes like LAMP2 or KCNQ1, you can see that gene therapy holds unique promise for these conditions because it offers potential to address the genetic root cause, not just the symptoms. Let's pause for a quick poll. Do you feel you currently have the knowledge and resources you need to confidently discuss gene therapy with your patients? Take a moment to answer, and we'll revisit this question at the end of our session and see how perspectives may shift. I'll now turn it over to Dr. Jonathan Schwartz to discuss gene therapy.
Thank you, Susan. So gene therapy is really sort of the epitome of patient-focused and targeted medicine. And it's really designed to address the underlying genetic cause of a disease rather than the symptoms, with the thinking being that if you can get a correct copy of the gene of interest into a sufficient proportion of the cells of interest or the organ of interest at a time before there's irreparable damage, that you can affect a very meaningful transformation or potentially even cure of an underlying disease. The advantages of gene therapy will include that in many cases it's given as a one-time therapy as opposed to a treatment that requires chronic administration. Um, and I think at present there's really a very strong proof of concept, particularly in disorders like spinal muscular atrophy, where gene therapy has enabled a growing number of young children with a very debilitating disease to live a vastly more normal and healthy life. Um, and obviously, each disease is different, each therapy is different, and there are going to be some conditions for which gene therapy is going to be much more readily applicable and more conclusively reverse the underlying disease. We're going to talk about one of those examples in a couple minutes. So moving to the next slide, we uh, really have two different overall approaches to gene therapy, one of which is in vivo, involving direct injection of viral particles. In most cases, this is adeno-associated virus, or AAV. And then one can also do ex vivo gene therapy, in which specific cells, usually hematopoietic cells or blood stem cells, are removed and transduced with a lentiviral vector outside of the body. Lentivirus is sort of a variant of a retrovirus, similar to HIV, but without the infectious capability. And then those blood cells are infused back into the patient and can then engraft in the bone marrow and produce healthy progeny, ideally for a patient's entire lifespan. We're going to talk about both in a little bit, but predominantly adeno-associated viral therapy. And this is illustrated on the current slide here. One can appreciate that adeno-associated virus is a virus that's naturally occurring in nature. It generally does not really cause infections or symptoms, but rather assists adenoviruses in doing what they do. Um, when injected into the bloodstream or other organ compartment, uh, the AAV finds its way into cells via different types of receptors on the cell surface. And then once inside the cell, makes its way into the nucleus, um, where it is then capable of not integrating its DNA into the host's DNA, but rather then generating copies of independent DNA known as episomal DNA, depicted in the lower right portion of the slide. And then that DNA is capable of uh, producing RNA and generating the protein of interest, ideally for the lifespan of the cell, and potentially in certain settings for the lifespan of a patient. Um, and this is what's known, this AAV has really become the predominant form of in vivo gene therapy for diseases of muscle and nervous system, as well as increasingly cardiovascular disorders. The benefits and risks are illustrated on this slide. Durability is something that's always in question, although I think increasingly we have a body of knowledge that is it suggests that especially in disorders of the heart, um, the durability is excellent and may be potentially decades, if not lifelong. Um, AAV has different tropism for different organs, with the liver being the greatest and the heart being also an organ where there's a significant degree of tropism. Um, and also, as I mentioned a few minutes ago, the potential for a single dose to provide lasting benefits. Nonetheless, there are challenges, and the biggest single challenge really, I think, has to do with the immune responses. Even though AAV is not a particularly highly immunogenic um, virus, because the doses that are required in order to transduce organs, particularly organs like muscles or heart, are pretty large. You can have potential for very significant immune reactions, and the field continues to evolve to try to figure out ways to render those reactions less problematic. And by and large, most of the other side effects all center around those immune issues. Um, moving forward to the next slide, this is the Rocket Pharma pipeline, which includes three lentiviral programs on the bottom portion of the slide for different hematologic or hematologic immunologic disorders, including programs in leukocyte adhesion deficiency and Fanconi anemia that are currently under evaluation by FDA and other health authorities for potential marketing approval. Fanconi anemia is a very severe bone marrow failure syndrome, and leukocyte adhesion deficiency is a very severe immunodeficiency. Both of these are frequently fatal in children during the early years or first decade of life. Rocket also has a program of AAV therapies that are directly injected for several cardiovascular disorders. And for the next few minutes, we'll talk about the program that's most advanced, that's currently in a registrational single-arm phase 2 study in Danon disease, and that's RPA501. And we'll talk a little bit about Danon disease and this program right now. Danon is an X-linked monogenic disease that's the result of pathogenic LAMP2 variants, lysosomal associated membrane protein. This is an X-linked gene that codes for a protein that helps lysosomes fuse and get rid of um, unwanted cellular material. It's sort of helpful to think of it as an integral part of the cell's garbage disposal system. Um, and senescent proteins, damaged mitochondria, any number of other cellular contents that are no longer useful in the cell need to be digested and extruded. And when LAMP2 is defective, that process doesn't happen as well. It's a process known as autophagy. And in the absence of autophagy, one sees a tremendous amount of buildup of autophagic or sarcoplasmic vacuoles. These can clog up the heart and cause extensive myofibrillar disarray. In Danon disease, this causes one of the most aggressive cardiomyopathies that's been described. It's aggressive in females and often fatal in middle age. It's particularly aggressive in males, and in boys and men, typically is results in a very severe, extremely hypertrophic cardiomyopathy that manifests during childhood and adolescence and is often fatal with a median survival of around age 20. Um, the female disease is a bit more indolent and a little bit more varied. Um, there are other manifestations of the disease in muscles and CNS, but these are not as profound and not as life-threatening. Heart transplant is potentially beneficial, but not necessarily curative. Um, Heart transplant is associated with tremendous complications. Patients, on average, live about 10 years after transplant. Uh, they require a tremendous amount of medications lifelong to prevent organ rejection, and there are many other long-term complications. Our program in Danon disease started with the most severely afflicted group, which was the male component, although plans are underway to initiate evaluation in girls and women as well. We targeted the therapy, both evaluating in our phase 1 study older patients who are considered adult or adolescent, age 15 and above, and pediatric patients in the 8 to 14 age range. Um, there were 2 different doses tested in the older group, a lower 6.7 times 10 to the 13 genome copy kilogram per kilogram dose, as well as a higher dose. And then that lower 6.7 E13 dose was also tested in 2 pediatric patients. We recently were able to update the results, which now extend to patients who've been followed for anywhere between 2 and 4.5 years, as presented last fall at the AHA meeting, and as also published in the New England Journal of Medicine. And we'll spend a little bit of time right now talking about those results. What we really wanted to first understand, if you can just back up a second, is we wanted to understand is whether or not we could actually transduce the heart tissue and whether that transduced heart tissue would generate LAMP2 protein. Would that change the structure and function of the heart? And would it actually benefit patients and could it be done safely? Um, with respect to safety, um, we did encounter some problems, particularly in the initial cohort, um, and specifically one patient at the high dose cohort, which is the middle column in the adult group, had a severe complement activation resulting in a condition known as thrombotic microangiopathy that resulted in severe kidney injury and thrombocytopenia. Fortunately, those were completely reversible. Um, But nonetheless, this is the sort of toxicity that we seek to avoid. We also found that in those first two cohorts, several of the patients had steroid exacerbation of the existing mild Danon disease skeletal myopathy. Steroids are used as part of the immunomodulatory regimen to try to lessen the immunologic reaction against the viral injection. In the pediatric cohort, we modified the immunomodulatory regimen to include a lower dose and lower duration of steroids, supplemented by the B cell monoclonal antibody rituximab, given 2 doses prior to therapy, as well as with sirolimus given orally, with sirolimus and steroids given for 2 to 3 months after gene therapy had been administered. And in that pediatric cohort with that regimen, we were able to treat patients without any severe side effects and without any drug-related severe adverse events. And that is the immunomodulatory regimen that we have continued to use in the current phase 2 pivotal study that's ongoing both in the US as well as Europe. From an efficacy perspective, on the next slide, we saw a very remarkable improvement across a range of parameters in 6 of the 7 patients treated. The 7th patient was treated at a more advanced stage of heart failure with a very diminished ejection fraction and didn't derive benefit and ended up requiring a heart transplant, which fortunately was successful. The other patients who were treated at a more moderate stage of cardiomyopathy progression have all benefited. Um, the 4 adult patients in that cohort are now all in their 20s, living independently, attending university, or holding down jobs at an age when most male Danon patients would have either had a heart transplant or succumbed to their disease. And again, all of these patients have been followed between 2 and 4.5 years. We saw remarkable reductions in the cardiac hypertrophy, as evidenced by reductions in left ventricular mass index, which is the first of the colored columns. The cells in this table are colored by the degree of improvement, with green indicating improvement, yellow indicating stability, and red indicating worsening. And as you can appreciate, there's not any worsening parameters. We also saw dramatic reductions, for the most part, in blood-based biomarkers of cardiac stress and cardiac injury, namely NitroProBNP and troponin, that's TNI. Troponin is markedly elevated in the serum of Danon patients. This is a test that's usually used to diagnose a myocardial infarction, and its marked elevation is indicative of the severe injury that occurs day in and day out in Danon patients. And so seeing reductions at times above 80 or 90% indicates that we're likely reversing a lot of the injury. All of these patients have gone from being symptomatic NYHA class 2 to asymptomatic, and patients have uniformly reported increases in the quality of life, as indicated on the improvements on the KCCQ score, which is the rightmost column of this table. Typically in heart failure or cardiomyopathy studies, an improvement of 5 points or more in KCCQ is considered a clinically meaningful benefit, and we've seen that across the board and much more in several of the patients. The next slide just gives you an illustration of the myocardial biopsies with several of the patients illustrated each per row. The top two rows are adult patients. The bottom two rows are pediatric patients. And what we're looking for is over time after treatment, whether or not they develop evidence of LAMP2 protein via immunohistochemistry staining, which is indicated by the brown staining that you can see as early as month 6 or month 9 in the second column from the left with that protein expression that's sustained in cases through month 24 and month 36 following therapy. On the next slide, we see that the LAMP2 staining is also accompanied by normalization of the myocardium, both on H&E tissue staining on the top row, as well as the electron microscopy sections on the bottom row. And in each case, over time, you're seeing reduction in those pathologic vacuoles as the cells are now able to undergo autophagy and digest all the discarded proteins and other material that they no longer need, with normalization of the muscle fibers within the myocardium. And so we were able to show this very conclusively, that this is working not only at the cellular level, but also with clinical parameters, as mentioned just a couple minutes ago. This slide just illustrates the design of the phase 2 pivotal study that's currently underway at several centers in North America and Europe. And the patients are treated with a single infusion with immunomodulation over 2 to 3 months prior to and after the AAV infusion. The primary endpoint is a composite, including both protein expression in the heart and a reduction in left ventricular mass index, with many other secondary endpoints across a range of imaging and blood-based and functional and patient-recorded outcome parameters. And that's sort of a whirlwind tour of our initial Danon experience. And this is really the first time that people have been able to show that gene therapy can potentially address a genetic cardiovascular disorder. In addition to the phase 2 study that's underway, as illustrated on the next slide, we have a multi-center Danon disease natural history study that is intended to really better define and delineate all of the critical signs and symptoms, events, and impact of the disease to better quantify the imaging and blood-based parameters and any number of other measures of the extent of the disease and how those progress over time. This is a study that's underway at a number of centers, again, in North America and in Europe. It's open to both male and female patients with Danon disease, and further information on this trial and where patients can enroll is available on clinicaltrials.gov, as well as in other material that we can make available. Obviously, it's sometimes an imposition for patients and families to enroll in a trial where there's not a therapeutic option, where there's benefit. But our hope is that this will enable us to optimally characterize many aspects of the disease that we continue to learn about and that we hope to be able to share with the broader genetic disorder and cardiovascular community. And with that, I'll turn it over to Kara. Thank you very much for your attention.
Wonderful. Thank you for the introduction, Dr. Manganaro, and for the background on the current Danon clinical trial at Rocket, Dr. Schwartz. As a genetic counselor, I will be talking through a case example and then a little bit of information about considerations for genetic counselors to be aware of in clinical settings with these emerging cardiovascular gene therapies, as well as considerations to discuss with your medical team, as you may have patients who are identified that could be eligible for trials. So to start with the case narrative, this is a 12-year-old male who presented with loss of consciousness and palpitations. He had a history of limited physical activity and learning difficulties, and his physical exam upon arrival in the emergency department was unremarkable. In the emergency department, they did an ECG that showed a shortened PR and delta waves, which is characteristic of Wolff-Parkinson-White syndrome. The ED discharged the patient and instructed them to have outpatient cardiology follow-up. You might be wondering what was done next and if other cardiac imaging was done. So in the outpatient cardiology follow-up, we'll see on the next slide that he did have an echocardiogram done that showed massive left ventricular hypertrophy. So the left ventricular end diastolic diameter was 45 millimeters. His left ventricular posterior wall was 12 millimeters thick, and his max interventricular septum was 11. So these are thickened, and I think it's pretty obvious actually in image B, directly to the left of where it says LV, is a pretty noticeable thickness that's not typical of what we see on an echocardiogram. So with this information, genetic testing is indicated. The patient now has a diagnosis of Wolff-Parkinson-White and hypertrophic cardiomyopathy. And a lot of people on this call who work in cardiology are probably familiar with this 2022 study. But if you're not, it's something that I wanted to highlight. And it's a study out of Northwestern University and with Invitae Genetics that assessed the uptake of genetic testing for cardiac diseases and the difference in diagnosis if a patient had a phenotype-based panel versus a more broad combined cardiomyopathy arrhythmia panel. If you ever used to order the prior sponsored testing, all of those combined panels were used to help create this study. And what this study found was that 14% of diagnoses would have been missed if only high suspicion cases had genetic testing. So if cases where the phenotype wasn't very clear, or if there was, say, the phenotype wasn't clear and there was concern about insurance or billing, we don't have all the details on what exactly a high suspicion might have been in the clinical setting, because as we know, we don't always submit every detail to the lab. But 14% of cases could have been missed with their genetic diagnosis. And then 10.9% of cases would have been missed on a limited phenotype-driven panel. So based on what we talked about at the very beginning of the webinar, with a lot of the overlap between HCM, DCM, ACM phenotypes, 10% of cases might not have had their diagnoses found. I know in the clinical setting, when we think about things like insurance coverage, access to testing, it's not always this cut and dry or this easy. But I think this is an important study to highlight so that we can think about if we should be ordering more broad panels on a consistent basis if you're not already doing that in your practice. So for this case example, the patient had a broad test, and that found, as probably not a surprise to you based on this webinar, a LAMP2 pathogenic variant. So this was a deletion at 257 that resulted in a frame shift and premature stop codon 26 amino acids downstream. So this mutation or variant in LAMP2 is expected to cause the gene not to form the protein or form a truncated protein that's not functional. So it's a loss of function variant, and loss of function variants in LAMP2 are known to be pathogenic and associated with Danon disease. So this result gives a patient a diagnosis of Danon disease. And as genetic counselors, I think a lot of us already know next steps would be family evaluation and further information. To revisit what Dr. Schwartz already talked about Danon disease, it's an X-linked dominant condition, and there's reduced penetrance and variable expressivity in women. For women with LAMP2 variants, you can have individuals diagnosed with a severe hypertrophic cardiomyopathy in childhood or teenage years, all the way through adulthood. So it is truly very variable in its expression. And that really highlights the need for family testing in these cases to know whether or not individuals need screening. As we already discussed, it presents with a severe cardiomyopathy that is often does not react to treatment and ends up necessitating heart transplant. And Wolff-Parkinson-White is often characteristic as a pre-excitation finding on ECGs. So if you have young patients with pre-excitation or Wolff-Parkinson-White as part of their diagnostic cardiology evaluation, something to consider is, could they have Danon? Currently, the standard of care for Danon is medical therapy for heart failure, often leading to the need for a heart transplant. And Dr. Schwartz already touched on the limitations of transplant as a treatment, which is something for those practicing in cardiology, I'm sure you are already thinking about. And we'll talk a little more about how to consider these types of different treatments, such as heart transplant, standard of care versus emerging therapies, and how to have these discussions in the next several slides. So now that we have the diagnosis in our proband, who is the 12-year-old male diagnosed with HCM in LAMP2, there was an uptake of family testing in this family, and the mother ended up testing positive, and on screening echocardiogram was diagnosed with HCM, and the maternal aunt also had testing and was positive and diagnosed with HCM. And these are examples where these relatives did not know they had HCM at the time of their testing or screening. And the proband really is what spurred this in the family. So I think this is close to home for a lot of us when we're doing genetic testing in the cardiovascular space and we find a proband. It can happen fairly often that when we test family members, we find a new diagnosis that we were previously unfamiliar with or did not know was there, because we don't always see symptoms in patients with these conditions until it's a very severe cardiomyopathy. So really to hone in now on why genetic counselors, why should we be thinking about this? This slide takes information from the ACGC practice-based competencies. So during all of our training at some point, these were the competencies used to develop our curriculum and used to assess us for our board examinations. And I just pulled some of the competencies that I think are really fitting for how genetic counselors are really appropriate healthcare providers to discuss gene therapies with families. We're providers that can critically assess genetic and genomic information and review medical and science literature to determine what the important aspects are. We can use our counseling skills to facilitate informed decision-making and lead difficult discussions with patients and families about what results mean and what different treatment options exist. We have background into research, so we can take the appropriate level of information to relay it back to the family. And we're really good at managing relationships between families and the care team. So we're really appropriate people to be helping bridge these conversations and navigate conversations with families as well as other medical providers. And at the end of the day, I think it's really important that we always keep in mind our goal as genetic counselors is to advocate for patients and families as well. Beyond ordering or helping people obtain genetic testing, I think a lot of us get into this due to the desire to help others, help patients and families. And this is really an opportunity for us to advocate for them, as there's a lot of families interested in learning more about gene therapies, and it's something that we can prepare ourselves to learn how to discuss. So how are patients identified for gene therapy clinical trials? This is an evolving area, and I think it's worth walking through the different ways patients are found historically as well as more recently. So I think we all know that the clinical care team can identify patients. That includes the genetic counselor, geneticists, cardiologists, nurse practitioners, and other members of the care team. So as we all listen into different webinars, talks at conferences, receive email blasts, we're pretty aware of different clinical trials that are ongoing for different conditions in cardiology and beyond. And if we have a patient who may qualify, we ourselves can identify them that way. But that's not always a sufficient number of patients that are identified to meet the endpoints of these trials. And there are other ways that this occurs. So genetic testing laboratories do provide significant data to help identify patients for clinical trials. And this is through communication that genetic testing laboratories have directly with the clinical care team, the ordering provider, direct communication between the genetic testing laboratory and the pharmaceutical company, and in some situations, direct communication between the genetic testing laboratory and patients. This is not strictly referring to sponsored testing. So any clinical grade testing, it depends on the lab, but in general, a lot of the main commercial labs that you all are probably familiar with do have a clause in their consent form that allows the commercial genetic laboratory to share the ordering provider's information of patients with pharmaceutical companies. So laboratories can share that, hey, Cara Barnett had a Danon patient. You can reach out to Cara and see if she is willing to talk to you about that patient. So if you are contacted by a company saying, hey, we heard you have a Danon patient, that is something that the laboratories have built into the consents. So when our patients are getting testing, that's actually something that when we're signing the consent form, we're saying that they're aware that can happen. Additionally, in the online portals that patients have access to, some of the laboratories that are patient-facing as well, when we order testing for them, if the patient goes in the portal to sign up and track their test, there's actually the option for them to opt in for direct contact from the lab about research. So patients also can be receiving direct communication from the lab that says, hey, you tested positive for this gene. There's this company researching it. Here's their information. So there are multiple ways that patients are receiving this information, both something they can pursue directly, as well as something that can be done with the care team as an intermediate step. Also, we all are aware there's a lot of really wonderful patient self-advocacy organizations that I know I rely on to help connect families to, and the families also rely on for information. So this is something where gene therapies are also discussed in these patient self-advocacy groups, both more formally on foundation websites, as well as even informally in Facebook groups. So there's a lot of different ways that patients are identified for clinical trials. And it's something where kind of being familiar with our laboratory consent forms and the terms types of data sharing happening can be helpful to know, okay, I'm getting contacted by this. Is this appropriate? Oh, yep, this is something that is in the consent form. Additional ways that genetic counselors are involved in patient identification, this is slightly more broad, but I think worth highlighting that a lot of times when we receive a genetic diagnosis, it depends on your healthcare system and what electronic medical record is used. But one of the things we think about is how do we code that diagnosis? And there's a lot of broad ICD-10 codes that exist for cardiomyopathies, and we have several of these listed here. Sometimes if we get a positive result, I know I've just put carrier of genetic disease, even if, say, there's not an exact specific gene ICD-10 that exists. There is a new ICD-10 for Danon disease that was approved in collaboration with multiple groups to try and help be able to stratify this diagnosis further for patients. And beyond just ICD-10 codes, even there's a lot of talk among the GC community of incorporating discrete data into the EMR so that genetic test results are more searchable. So when clinical trial sites are developed, something that the site PIs might be doing or talking about with their clinical collaborators is how can we mine our EMR data to try and find possible patients that could be eligible? Besides just the spreadsheets that a lot of us keep with results, what other ways can we look? And some of those are things like discrete data, including ICD-10 diagnosis codes. So when actually discussing clinical trials, what questions should we be asking? These are a lot of broad questions. I'm not going to discuss any specific trial, but rather I wanted to provide the questions that we should be thinking about so that if these situations, and really when these situations become applicable to you, what should you be looking for? The top two things are typically, what's the criteria of the trial? Does my patient even qualify? And who is the sponsor? Identifying this information can help you then answer the next questions. So what is the inclusion criteria is typically available on clinicaltrials.gov, and the industry sponsor is listed there also. The industry sponsor website typically includes further information as well about the trial and contact information for individuals as part of the trial where you can get more information. To find information about the science behind the trial and what preclinical data has been published, PubMed is a great place to go. Some of this may also be on the industry sponsor as well. More broadly, when we're thinking about discussing with patients, it's important to know what are the benefits and risks, what are possible side effects. Dr. Schwartz talked a lot about some of those immune side effects patients can experience, as well as does participation preclude the patient from possible future treatment. So typically, patients cannot receive an AAV more than one time because of this immune response. That could change in the future as different ways of administering gene therapies develop, but it's something to think about where if they have a gene therapy now and it prevents them getting one later, what is the right timing for this patient and family? What are the benefits and risks to them if they want to proceed into a trial now, if they're really excited about it, versus what benefits might there be from waiting? And then talking to peers and colleagues is always great to see, can you find other individuals that are connected with the trial? And to do that, you can go to clinicaltrials.gov. So as I mentioned on clinicaltrials.gov, there is the inclusion criteria listed as well as exclusion criteria. So you can look there directly to try and see if your patient is someone who would qualify. And then you can talk to your cardiology team about this further if you need to discuss things such as like specific echo findings. Another part of clinicaltrials.gov is the location sites of the clinical trials and the contact information for the site PIs. That is something where you can contact site PIs directly if you have patients who might be interested or if you have questions about the clinical trial. So there is contact information listed as the main contact for that site for all of the clinical trial locations. This is uniformly present across clinical trials on the website. So you should feel empowered if you have a patient who's really interested and you don't have all the answers from your medical team or from your research. You can contact site PIs to ask to talk to someone on their team for more information or just to connect the patient to them directly. So what other information should we discuss more directly with the medical team? Some information to consider would be, what are the obligations of participating in a clinical trial? We want to make sure we can convey to the patient the number of visits this might require, what investigations are done. Is it echoes? Is it MRIs? Would they prefer or do they need a cardiac biopsy? What is the monitoring timeline? How long is the trial going to last? And how often are the follow-up visits? Additionally, trials are not located at every medical center. So it's something where we need to know, can the patient travel to the location of the trial? And can they commit to the long-term follow-up needed to measure if we're going to meet our trial endpoints? Once the trial is over, what type of monitoring should the patient still have? When we're thinking about cardiomyopathy, it's probably something where, the patient's not just going to be discharged from the cardiac clinic because they received a therapy and say they had a positive response. We can't just not continue to monitor them. So something the team needs to discuss is, once the trial's over, how will we continue to monitor them? What will their standard of care be? Additionally, in general, will these gene therapies help my patient? For the patient, depending on their level of disease progression, is the gene therapy better than their standard of care treatment? Does it offer better hope than what the standard of care does? We have already talked a lot about how in Danon patients, a lot of people believe this is true, where in Danon patients, you have rapid progression that often necessitates a transplant. So is this something where, do we believe the data that's coming out? Are we confident and comfortable pursuing a trial? If the trial doesn't help me, will it impact my ability to be in future studies? What is the cost of the trial? What does the sponsor cover? What might my insurance cover for me to participate? These are all questions that we as genetic counselors can help try and find as many answers as we can ahead of time to prepare for some of these conversations with parents and families that might see information about these trials and be interested in learning more. So as the final slide for me, I have this additional resource that was coordinated by the SADS Foundation. So completely independent of our webinar, Dr. Landstrom and the SADS Foundation put together a webinar mostly targeted at patients and families for what to know about clinical trials. And he talks more broadly about the different phases of clinical trials, what they mean, and what questions to ask. So I think even as clinicians, it's still a helpful webinar. The QR code here will link you to it. It's available on the SADS Foundation YouTube channel. And it has a lot of great information, as the SADS Foundation always does if you're not already connected with them. Thank you.
Thanks, Cara and Dr. Schwartz. That was excellent. So I asked you at the beginning of this session this question, and now we'd like to revisit it. Do you now feel more confident in your knowledge and resources to discuss gene therapy with your patients? Whether it's your first exposure or you've been following this field for years, we hope that today's webinar gave you the tools and framework to feel better equipped. And if there's still areas where you want more information and training, we'd love to hear that feedback as well. We're looking to develop more content. We're having a second webinar coming soon. As Cara discussed earlier, having an accurate diagnosis is really critical to help identify patients and potential candidates for clinical trials. Rocket supports a sponsored genetic testing and counseling program for those individuals living in the US with suspected inherited cardiomyopathies, including those who have a family history of Danon disease or a clinical diagnosis of cardiomyopathy not explained by acquired causes, or those meeting a combination of red flags listed here. You can scan the QR code on the slide for more information. If a proband tests positive for a LAMP2 variant, you can easily refer them, their relatives, for cascade testing through this platform. There's no time limitation, and as Cara had discussed, cascade testing is really important, especially to be educating your patients and their families in these types of disorders, especially where they're X-linked and they can have a heterogeneous clinical presentation. What's especially valuable, I think, about this platform is that it can support remote cascade testing for at-risk relatives, even if they're outside your clinic, your practice, your state. The program has the option as well to include genetic counseling if you're not able to see the family member yourself. Thank you again so much for joining us. As we've seen throughout the session, genetic counselors are essential to the success of gene therapy programs, not just in helping patients understand results, but really identifying candidates, providing them with resources, access to clinical trials, and help really bridge that gap between science and care. So whether you're working in a high-volume cardiac center, a regional genetics clinic, or even on a virtual counseling platform, your role is ever expanding from diagnostic navigation to therapeutic guidance. And before we close, I really want to highlight one additional opportunity where you can help make a real impact. As Jonathan spoke before, we have a Danon disease natural history study. It's an observational study aiming to capture long-term real-world data on how the disease progresses over time. And referring families to a study like this is really a meaningful way to help support those who might not yet be ready or eligible for an interventional study, but want to still participate and help in advancing the science. So we hope today's discussion gave you some tools and confidence to recognize when a gene therapy might be appropriate, to help counsel patients and families about evolving treatment options, and help them make informed and empowered decisions. On behalf of Rocket Pharmaceuticals and our speaker team, we want to thank you for all the work you do and look forward to continued partnership as gene therapy reshapes the potential future of inherited cardiomyopathies.